everyone to the afternoon session. We have 3 talks this afternoon. The workshop is evolving towards an informal format, so also our speaker agreed to be interrupted during the talk, so if you have questions, feel free to ask. So I'm very happy to introduce Anatoly Kolomeisky from RISE, and he will tell us how to find targets that are always hidden. Please.
Okay. Thank you, organizers, for gathering such a group of talented people who presented excellent research. And it's better 3 years later still to meet in person. So this is probably the longest title that I ever had in my research. So anyway, so let us start. So as you know, everyone knows, so one of the most important biological processes is the transfer of genetic information. You need to read what is written in DNA, transfer it in RNA, in proteins to fulfill its biological program. And the initial step of this process is when several classes of proteins known as transcription factors, they bind to specific sites on DNA. This is known as the promoter regions. And when they bind it, RNA polymerase can create proper initiation complex, and you can properly do RNA transcription. You can properly copy the information. This is a part of so-called central dogma in biology. We know these things here. But then when we come to real life, there are complications. As you know, in eukaryotic cells, DNA are tightly packaged. In order to fit in relatively small cellular volumes, DNA should be packed, compacted with several different proteins, multiple proteins. Mostly it's nucleosomes. And we actually saw today this essentially, we saw these chromosomes here. And although there is still controversial what's going on between this and this, we do know that DNA is compacted, bound to nucleosomes. And the problem comes is then if DNA is compacted, and the data shows that up to 90% of DNA typically cover it, how you can activate the genes if it's always closed, if other proteins like nucleosome prevent it, what to do? Fortunately, nature solves this problem by using several classes of these transcription factors, which are known as the pioneer transcription factors. And it is known that they are able to penetrate the chromatin and somehow activate those genes which are closed. And there are several families of these so-called pioneer transcription factors are known. And again, pioneer means they first open chromatin, and then other transcription factors come and do the work. So that's what they call pioneer. They're the first. Why we do know that they opening somehow chromatin structures and go inside, unfortunately, we don't know much about the mechanism. So how exactly the transcription factors can reach a specific site is not very well known. It's not clear. So, and this is the story that I want to tell you about possible things, how it might happen. But before proceeding, I want to remind you that there are other, I call them normal, but just the non-pioneer transcription factors, those that start to work after pioneer open chromatin. So we know more or less, because this was a topic of research in the last 50 years from both theoretical and experimental point of view. And in this audience, there are several actually experts who did the work here. And this is also related to the work on homology search, which was nicely presented a couple of days ago. So we more or less know that if you do in vitro measurements on this normal, non-pioneer, the proteins actually find, in many cases, target faster in comparison with idealized Smoluchowski case. With the idealized case, if there is no DNA, you would have one specific rate predicted by Smoluchowski rate, but the presence of DNA, the fact that protein can sometimes bind to DNA scan and also intersegment transfer makes process faster. And the proteins can find targets faster. So we more or less understand that this is known as the facilitated diffusion mechanism, and this alternation helps. And again, as I said, the mechanism here is more or less understood. This presence of DNA, intersegment transfer, allows effectively increase the mobility of protein, and that's how you can beat Smoluchowski limit. But this is in vitro, and of course, if we want to understand what is in vivo, it's not clear how you can extend this picture. So essentially the question is how pioneer transcription factors can do these things here in vivo if they're not able to follow, it seems that they're not able to follow this mechanism. Good. So fortunately, there are experiments started to appear in recent years, and these experiments gave us some clues of what might happen. And I mentioned one experiment, but there are several of them came out. So what these experiments measure, they measured simultaneously kinetics of association and dissociation of pioneer transcription factors. The first two are pioneer, and non-pioneer transcription factors, which come from the same organism. I think it was from yeast, if I'm not mistaken. Anyway, so they measured this kinetics of association, dissociation. There were a couple surprising observations, and let me try to emphasize them. So the first one is that association dynamics seems to be similar for normal and for non-pioneer and pioneer transcription factors. In other words, normal, non-pioneer, and pioneer transcription factors bind with approximately the same rates to naked DNA segments, to free, and also they bind with the same rates to nucleosome-covered one. Of course, the rates to come to nucleosome-covered is much slower, but between each other, they're more or less the same. The difference between pioneer and non-pioneer transcription factor comes in dissociation. They dissociate differently. So it seems that pioneer transcription factor are able to stay on nucleosome longer, while normal kicked out quickly. And then the situation is vice versa at naked DNA segments. Non-pioneer can stay longer, while pioneer are kicked out. So the difference comes in this dissociation dynamics. Okay? So this gave us a clue that maybe we can do something, try to model these things here. And then, so if you ask people in the field how pioneer transcription factors can find it, you can ask biologists, they can typically tell you, oh, it's easy, it's nucleosome breathing. So in other words, nucleosome, what I told you, it's not a frozen structure, right? So there is a nucleosome is breathing. Imagine this is DNA and there's nucleosome, it's breathing, so it's opening. And several years ago, people started to prepare quantitative models because there is more, you have transition rates, some of them. People can start to try to quantify this idea of that nucleosome breathing is something that what is needed. So the idea the arguments were if you sometimes unwrap, then you liberate the sites and then protein can come and activate gene. So there were the models, and that's exactly what I'm saying. So occasionally, when you unwrap, you are opening sites, and transcription factor can come and bind to these things here. It's a nice model, but there is a problem. So this is what I always like to have models which have numbers. I prefer to have analytical approximate model to any simulations because you cannot dispute if something wrong. Numbers. So when we put the numbers in, there is a problem. So nucleosome is quite short. It's in the order of several microseconds. But the problem comes for those sites which are buried inside. So remember, I'm not doing like this. I'm opening only partially, right? So while I can probably cover sites over there, sites which are deeply buried are not reachable. So, and then if you put numbers in, this is estimate from these theories which use experimental dissociation association rates. It's 30 hours, it's too much. So clearly, this mechanism is not wrong, but it cannot present the full picture. So there is something else should be added to this picture. Yeah, it's an excellent question. So first of all, there is still controversy how much you open. I talked with several people. Some people say it's open up to 10 base pairs. Some people say up to 70. But none of the experiments I have seen can open 150. To remind you that the histone is wrapped around approximately 160 base pairs. So this is a problem. Yeah. And it's exponentially decreasing with energy, so this is a problem. Good. So we are proposing possible mechanism, but I want to emphasize again, it's a theoretician's presentation, which means I might be completely wrong, and we know in biology we were wrong many times, but this is something which is, I think, is plausible. Good. So, and the model that I will present is stimulated by this observation. So for some pioneer transcription factors, I want to emphasize only for one that I found so far, people structurally see similarity between pioneer transcription factor, in this case it's called FOXA, and linker histone. In other words, there is a similarity in structure between pioneer transcription factor and histone, or parts of the histone. Okay? This similarity gave us the key, so maybe this is what allows them to come and access. They can remove part of the interaction between DNA and nucleosome, substitute with their own interactions, and that might be the mechanism how it might work. So this is the idea, and we call this idea interaction compensation mechanism. And the idea is, as I told you, there is a way the pioneer transcription factor can bind by displacing some of the interactions which exist between DNA and nucleosome with their own interactions, and this can allow them to enter. And this is based on some structural information. And we can build the model. And by the way, this is a picture, very naive picture, how it might happen. So imagine this is your very crude DNA and histone, right? And this is your pioneer transcription factor comes, and somehow it displaces interactions and gives and substitutes with their own interactions. And it allows pioneer transcription factor to stay longer. That would be consistent with experimental data. It's hard to come, but it's harder to leave. They're staying longer, pioneer transcription factors. In contrast to normal non-pioneer transcription factors, which do not provide such interaction. So this is the idea. You can create a model. You can do the stochastic model or chemical kinetic model. So in this model, you use association rate to bind to naked or covered region, and the prime corresponds to binding to nucleosome covered region. But there's additional things in this model which, so these association rate dissociations were measured, and I know order of magnitude of these rates, but we have additional things in the model. We also assume that there is a sliding is possible. Sliding is possible in the naked DNA as long as the nucleosome covered. This was not experimentally observed. So this is, again, this is important graph because it emphasizes the difference between pioneer and non-pioneer transcription factors. So both of them binds equally well to naked, this is binding to naked DNA. You see this barrier, the same for blue and red. For blue is pioneer, red is normal. The same, so it's the same rate of binding, but dissociation is different. Normal stays better bound to naked DNA, but pioneer is not. And situation is similar over there, although the rate of binding is much slower. But both of them has similar rate. Both of them has difficulty to bind to nucleosome. We understand why, right? You have to break some bonds. But due to this interaction compensation, pioneer are able to lower the energy, and so they dissociate slower. So this is, the whole idea is over there. And you can try to put it in the numbers. You can assume something about was a association rate. You can assume something about dissociation rate to reflect these experimental observations. This is experimental observations. And you can say something also about, you can postulate about sliding. You can argue that maybe pioneer slides a little bit faster on nucleosome in contrast to normal. Rates anyway are relatively slow, but you can postulate it. And you can solve the model exactly analytically. This is another advantage of this. If something wrong, you can see what it's wrong. And this is the graph. So the graph shows search time to find the target in my system, which consists of nucleosome-covered DNA as well as naked segments here, right? So let's start. So the red corresponds to non-pioneer, normal. And you can see that normal can find targets better if targets are located in the free regions. This is expected. Normal transcription factors are pretty good. But in contrast, blue is that pioneer are not so good. But the story changes if you go to the covered region. So this region corresponds to covered, nucleosome covered region. You can see that normal is really blowing out with the same effect that essentially we discussed a few minutes ago. And please note this is log scale, so it's a really huge slowdown, 3-4 orders of magnitude, while pioneer is doing better. It's not perfect, yes. Time. mean search time or mean first passage time to find the target if you start from a solution. Yeah, you start in a solution, and you stop your process when your protein binds to specific sequence, to target. And by the way, in this lattice model, each of the sites corresponds to 10 base pairs, which is a typical size for proteins to bind. It's an order of magnitude, but it's probably okay. Right. Okay, so you can see that pioneer transcription factors are not perfect, but much better, several orders of magnitude, and you can quantify it even using the heat maps. So the heat map shows this is a position where nucleosome starts, and this is a position of target site. And essentially, you can see there is acceleration up to 2 orders of magnitude in diagonal, and diagonal corresponds where your nucleosome usually. Towards your nucleosome, pioneers doing much better, 2 orders of magnitude, up to 2 orders of magnitude better. And of course, it's relief from 30 hours to go to 20 minutes. That's what the prediction. And it's interesting, it's comparable with the time for homology search that you report. Probably it's a coincidence, but it tells me this is a reasonable time scale. Okay, in 20 minutes, it's okay if I do it 20 minutes, but 30 hours is not okay. So this is the story. And again, so let's try to understand why it works. So this is a keywords, interaction compensation effect. So the pioneer in our picture probably can bind to this histone-covered DNA by displacing some of this interaction and substituting with their own. But I want to emphasize again, this is one of possible microscopic pictures. You might come out with other things that protein can bind to nucleosome. Yes?
How long is the DNA you're considering?
Yeah, so in these specific models, we took it essentially 600 base pairs. But the results do not depend much. You can do it shorter a little bit, right? Okay. Yeah, so these arguments explain why pioneer transcription factors do not want to dissociate, because they stay bounded. And it seems natural to propose that nature may be tuned kinetic properties of these transcription factors to work exactly like this, to work in the nucleosome, to go inside of nucleosome. It's not good to be on naked DNA, but to enter a nucleosome, you have to do something. And maybe nature tuned the kinetic rates to do it. Good. So this is my naive explanation of compensation mechanism. So if you want to go to supermarket and get something, you need to have a compensation. And if you do not have, you might be in trouble. You will be kicked out and maybe even worse. This is actually Houston Police Department picture. Anyway, doesn't really matter. So they really have these hats, if people are wondering. Anyway, so okay, good. So this is nice, but I forgot to mention important things that in this model that I was doing, it was frozen. It was frozen, nucleosome didn't move, but nucleosome breathing is real. It does happen. So what it is, so, yes?
Is it one, in your model, you're imagining one pioneer?
That's correct. In reality, will be many, but because they act independently, you can multiply by the number, you can get concentration. This is a typical argument. You can go from single particle description to bulk description. If each of these agents acts independently, you just multiply it.
But do we know that they act independently? Or
I don't. I assume the simplest possible things. Maybe it's not. But in the simplest possible picture, I assume they act independently. Good. So we can extend the model because it will be more states, but still manageable. You can solve it still analytically. And you can calculate, so what will happen, how the nucleosome breathing will affect target search dynamics. And results are actually unexpected, something that we didn't expect and spent 3 months. I thought my postdoc is wrong, but essentially I was wrong. So this is, let me explain the things, and you will see it. So let's start with the non-pioneer transcription factors. And this search time is a function of target position. And on the top, this black one is search if I'm always fully wrapped, if I always like this. Then the bottom, the blue line, is if I always partially unwrapped, not fully, but partially unwrapped, frozen. And the red curve, if I have this. And I get the results which is expected, right? I was expecting something in between these two limits, and I got something between these two limits. Now I go to pioneer transcription factors. And again, remember, this is the numbers for association dissociation which come from experiments. It's not, I didn't place the numbers. And there is a surprise. Again, this is for fully wrapped. always like this. Blue one for always partially unwrapped. But when you turn on fluctuations, you can do things better. You can essentially, you can find things faster, especially in this covered region. This corresponds to covered region. So this is something counterintuitive. When you do the fluctuations, your sites in fluctuations are more frequently covered than it's always partially unwrapped. How you can do things faster. Okay, this is the reason we spent 3 months with the postdoc trying to figure out, we did simulations and analytical results. So we have, so this is the surprising observations. And again, it's only in nucleosome covered region I can do things better. But in other regions, it's a comparable rates as with a partial unwrap. So how to explain these things? And by the way, this is a way to put it in numbers. So if I compare, so this acceleration, if I compare only binary transcription factors with itself for different positions, so this is essentially on naked region, but this on covered, I can do things better, up to 4 times better, if I can trust my numbers. But in comparison with binary transcription factor, I can have a huge effect up to 35, which is a huge effect. So how to explain these things here? So we didn't come out with anything simpler. We have these phenomenological arguments. Let me try to explain to you. So let's look at the macroscopic picture, okay? If from macroscopic point of view, so this on the left, this is binary transcription factor. From macroscopic point of view, I can identify like 3 big states. My protein can be in solution, free. My protein can be bound to partially unwrapped conformation. My protein can be bound to fully wrapped conformation. And given the rate that I mentioned to you, the most probable event is that you go and bind this one. It's very rarely you bind here. Remember, it's very difficult to associate to nucleosome covered region. Yes?
I have a question. Could it be that pioneer transcription factor, when they bind, could they affect actually the open state of the breathing nucleosome, like to make it longer, last longer, or to make it open wider?
Yeah, right. So you're talking something what generally called cooperativity. This is possible. But we decided, it's not in our model. The model doesn't have it. And this is exact analytic result as well as simulation. So it is possible, maybe, but I don't have any experimental data. And the models that I told you has nothing. Like, there is no cooperativity. OK, good. So let me emphasize again. So it's a preferred changes. It's not, occasionally you can bind like this, but preferably you go from solution to partially unwrapped because you go to the naked region. And then fluctuations happen and you go to fully wrapped conformation. So this is a topology of this macroscopic transitions if I have pioneer transcription factor. If I go to non-pioneer, it's a different topology. I have a solution state. From a solution, yes, I can bind to partially unwrapped conformation. But then this conformation happens, but I dissociate it quickly from fully wrapped. So there is a different topology of transitions here. Let me try to maybe explain this a little bit better also this way. So imagine, this is my pioneer transcription factor, and I have this partially unwrapped conformation. I can bind here reasonably well. Both of them can bind to naked DNA reasonably well. And if I bind here, and if there is no conformational changes, I will be quickly kicked out because pioneer transcription factor does not like naked DNA. What saves pioneer transcription factor is these fluctuations. These fluctuations which wraps it, saves it, because now you have a chance to create these interactions which stabilize you. And so this is a mechanism of this very strange mechanism from my point of view when this conformational changes can accelerate finding the targets here. Good. So yeah, when you do the theory, you have to be always very critical. Otherwise, you can sell air. So yes, it seems it's a reasonable picture, but there is a lot of if, okay? So first of all, we postulate the existence of sliding of rates. It was not observed. Sliding was not fast, and I could argue, if, maybe sliding rate is so slow that they were not able to do it experimentally. But that has to be checked. It might be it doesn't exist, and then you have to change the model, or you have to do something. Then, again, in a typical theoretician way, DNA for me is homopolymer, but it is not. It has sequence, and it's known that the nucleosome breathing, for example, is strongly sequence dependent. It's not in the model. Will it affect it or not? I don't know. Then, in addition, some will already mention this nucleosome breathing is a complex cooperative process, okay? I really treat it as a very simple breaking bonds, but it's not. It's much more complicated, and people argue that it's asymmetrically breathing, so when it starts to unbind from one side, another side gets actually stronger interacting, and so on. It's a very complicated free energy landscape, also which coming from experiments as well as from MD simulations. Now, there is additional things here. As you probably know, chromatin is much more complicated. I completely neglected the existence of other players, like chromatin remodelers, right? Chromatin remodelers also can change the mobility of nucleosome. In addition, nucleosome also slides. Not as fast, but it can also slide. It's neglected. Is it important or not? I don't know. I could argue that it probably would not change the physics, but it will change the numbers, definitely. So that needs to be checked. Good. So this is my conclusion. So I presented possible microscopic, microscopic is too strong word. Mesoscopic mechanism, because I still don't have much microscopy here, right? It's a possible mesoscopic picture how it might work, how pioneer transcription factors can actually enter chromatin, how we can find targets which are hidden almost always. And the key observation or key postulate is this interaction compensation mechanism. So, and according to this mechanism, I argue that when they come to nucleosome, it displays some interactions and substitute with their own interaction. But I emphasize again, specific details I don't know. I propose one of possible picture based on structural information, but it could be several of them together or something else. I don't know. So essentially, but the good thing about this, you put the numbers which come from mostly from experiments, and you can see the significant accelerations. You're getting the numbers which are reasonable, okay? And it's not a simulation, it's analytical theory. And if this story is correct, you actually have a beautiful example of something how nature can solve a complex biological problem. So we postulate that it might tune these proteins to do things faster, to find the hidden targets faster, using two aspects of interaction compensation. You entering better to nucleosome covered region, staying longer actually. You're not entering well, but you're staying longer there, allowing you to search better. And you also take advantage of nucleosome breathing. Another interesting observations here. Good. So this work was done by my talented postdocs. And this is the funding. Yes, and the Welsh Foundation, its money in taxes comes from oil. Doesn't matter. Good, so, and thank you. I'm happy to answer questions.
Thanks a lot. Students first. Good.
A question about cooperativity. So this is basically just thermodynamic consistency, right? Because if you have a higher free energy of the transcription factors on the wound state, then it's going to tend to cover those regions of the. I mean, if the theory is thermodynamically consistent, you need to couple the binding to the DNA and the wrapping.
No, this is consistent. When I talked about cooperativity, it's something more difficult, I would say. So it is known, or at least experiment shows that when nucleosome breathes, so it opens first segment, and second is easier to open, third is easy, but then again it's harder. So there is a cooperativity. So the rate of opening next segments depends how many already open. In this sense, cooperativity, you understand, right? The theory is thermodynamically consistent this way. The rates on-off
in the sense that when the pioneer transcription factor is bound to an open region, that region tends to close because of.
That's right. Yeah, this is
more than it would close
yes.
otherwise.
The numbers that I use are coming from experiments. So I more or less sure this is correct. What I talked about cooperativity, it's a next level effects, which is not here.
Anatoly, so these pioneer transcription factors and the normal transcription factors structurally are similar, are very different. I mean, could you mutate one normal transcription, one pioneer transcription factor by mutation, some amino acids, and get normal one? I mean, what is the structural relation between them?
What I know, and I don't know much, they're quite different structurally. But there are no structures for all of them. For normal transcription factor, we know structures, but for pioneer, only few structures are available.
Besides the structures, I mean, at the level of, I don't know, charge, polarity, non-polarity, amino acid content, I mean, are there chemical differences between them that may explain why some of them have these properties respect to the others?
Nothing that I know. The only thing which I told you was this similarity with histone, linker histone. That's the only thing which I found, and the only thing which came to my mind. Do I have a structure here? Yeah,
I see.
this one. This is the only thing that gave me the idea, but I don't know. But I said there is not enough structural information of them.
Okay, thank you so much.
Tolya.
Sorry.
I mean, are there other, analogous mechanisms in other systems, for example? I mean, I know that people have found things like nucleotide binding depends on nucleotides, so that where you've got one molecule displacing, where, you know, one molecule is actually displacing another one because it has a shared binding site. So, you know, this mechanism, I mean, it should be all over the place. And I just wonder, is it, I mean, are there other systems that use this, you know, this kind of, you know, it could be strand displacement with DNA binding or something. I mean, it seems that this would be a very general mechanism. There should be other examples of it.
Could be. The only thing I can tell you, something related to experiments that Anjana was showing, homology search is something like this also in some sense. Probably not enough quantitative information to pin it down, but I do believe that you're right and then something like this is happening. I remind you, it's a mesoscopic model. I still don't tell you the details how it's happening. I don't know.
The nucleosome breathing, is there periodicity to it? Like, is there rhythmicity to how it breathes? And
yeah, yes, it is. So it's depending on the system, depending on all the sequence, underlying sequence is of order of 1 to 10 milliseconds. And there's actually interesting catch. Almost everyone doing in vitro experiments on sequence called 601, or Widom sequence, Jonathan Widom sequence. But in nature, such sequence doesn't exist. So 601 is very strong, and you can make it very strong, and breathing is less because it's very stable. But in real in vivo systems, sequences are such that breathing is much more frequent. Okay, did I explain things? So you have to be careful what you're looking. In vitro sometimes give you results which are do not correspond well to what is in vivo.
because in going with, following up on Joe's question, if nucleosome breathing is a more general phenomenon across domains of life, then could there be breathing even on bacterial nucleoids, which allows then some transcription factors to access the DNA faster than others?
I didn't think about this, yes. I don't know much, believe me. I'm still a chemist, sorry. So possibly. As you probably know, the biggest driver for theoretical people is excellent experimental results like you're producing. Quantitative experimental results, then you can do something theoretically. Otherwise.
That's very cool, thank you.
How is the behavior of the average time to reach the target as a function of the breathing frequency?
Okay, yeah, so.
Is it non-monotonic?
It's, so the model that we had was simple two-state model. You could do more complicated model with intermediate breathing. And so we took the rates from experiments, more or less, right? But of course, if you are very rarely fluctuating, you're close to over there. If you very frequently fluctuating, you're close to over there. There is some optimality, I agree.
Okay.
And
So
it seems that
plot,
experimental things close to optimality, but
so there is a maximum or a
it's
minimum.
something like this. So in any way, for all possible rates, this is a common phenomena, but the amplitude of this phenomenon depends on the frequency of fluctuations. You are right.
Thank you.
Here, regarding at least the role or the importance of cooperativity, have you tried to run experiments with these sequences that are more histone prone and without? So, because if you increase the affinity for a histone, then you increase the probability for formation of a nucleosome. And if you think that it will be there most of the time, then eventually the cooperativity will not be so important, right? So I don't know if you can really have a measure, but at least you will have an answer to whether it is or not important for the model to see things or not. Have you tried to do?
So first of all, I'm not doing experiments. I'm very bad in this, believe me. I can do only some calculations. I don't know if they did these experiments, but again, I want to emphasize the value of such theories is because they can be tested, because they're quantitative. There are specific quantitative prediction, you can test it, and it's wrong, then it will stimulate you to do something better. Okay? So at this moment, I believe this simple model describes all available data that I know reasonably well. Yes, yes.
So can I understand this effect by imagining that it optimizes this trade-off between the number of rounds to find the DNA versus duration of a given round, right? Yeah. So that's sort of this typical explanation of why facilitated diffusion, 1D diffusion can help, right? Because it reduces the number of rounds, but each round becomes longer. And so is there something similar going here? I mean, the basic question is, if you're right, your K-off is also lower, I guess, when you're stuck at the wrong histone, no? So, right, so I guess it can help when you're on the DNA that's covered by the histone, where the target site is, but you can also bind to histones that do not contain your target site, and then I guess you're longer stuck there, or not?
Okay,
or
so it still follows these multiple rounds, right? But I believe the most important effect, not this one, but let me try to explain again. So from a solution, I mostly typically come to this naked region. It's easier for me to come for pioneer transcription factor. Without conformational transitions, I will be quickly kicked out. I don't like to be there. It's easy to come, but you can go quickly. What saves this protein to be in a system is conformational change. It's immediately from partially unwrapped, it goes to fully wrapped. And fully wrapped is good for this protein because it starts to create these interactions. If it starts to create this interaction, then it allows to go inside and find the targets. That's the way to do it.
Exactly. So if the target is there, it makes it better to find that target.
Correct.
But when it ends up inside a nucleosome that does not contain the target, it spends also more time there, right? That's
right.
bad,
That's bad.
Right?
That's correct.
And so you have this trade-off still.
That's
right?
correct. That's correct. This is I understand your question. I didn't investigate this, so so it's idealized picture, assuming that there is a target somewhere here. Yes, you're right. I didn't. You're right.
More questions? If not, let's thank the speaker again.